Hi, thank you. So my name is Nikki Watson, and I am the core facility manager at the Keck facility at the Whitehead Institute and the electron microscopy specialist. This image was generated with work done with Mary Gering's lab, and she works on the effects of fertilization and seed development, the molecular effects of fertilization seed development on Arabidopsis. Arabidopsis is from the family Brassicaceae. Is related to the mustard plant that you see in California on the yellow hills. It was discovered in the 17th century in the Harz Mountains of Germany. Earliest reported mutant was created in 1873. Chromosome number was first determined in 1907 and became a model organism in 1943. So this little plant has been around for a very long time and has sponsored a lot of great careers in science. So the Gering lab it studies how plants make seeds. So let's review our Botany 101 and remember how plants make seeds. So the basic common flower parts, we have petals to attract the insect. We have the stamen and the anther where the pollen is generated. We have the stigma, the pistil, and the ovary where the seed is developed. So the anther releases the pollen. It gets placed on top of the stigma by wind, by bugs, by whatever means. Travels down the style, meets with the eggs, and develops in the ovary into a seed pod or into a fruit. So Mary's lab works with Arabidopsis Lareda and Arabidopsis thailandia. These species are very closely related to each other, but they reproduce in very, very different ways. In particular, Arabidopsis Lareda cannot self-pollinate, and Arabidopsis thailandia can self-pollinate, which sounds like a small difference, but it's a big deal when you're one of these plants. So we imaged the flowers of both these plants and compared their morphology and their anatomy. And that's where we came up with this picture that was then submitted for this great event today. This is Arabidopsis Lareda. You can see the stamens here have not yet matured enough to release their pollen. However, the stigma here is mature enough to receive the pollen. So if this plant is going to produce seeds, the pollen must come from some other plant, from some other place. Also, it has the capacity to kill its own pollen if it happens to get on here. While Arabidopsis thailandia, if you can see the purple dots on here, the pollen grains have matured, they've exploded, they've come out. They're actually landing on top of the top of the plant here, so this plant can, in fact, both sexual parts of the plant are mature at the same time, so it can self-pollinate. So that was sort of the take-home message of this image. We can take advantage of these observed differences and make evolutionary comparisons on the process of the fertilization of seed development. So since I'm an electron microscopy specialist, I can't leave the stage without talking about producing these images. Scanning EM is not a difficult process to master, but it does have to be done particularly in a particular way. So first we collect our material, we chemically fix it, fully dehydrate it, that is, replace all the waters in the cell with ethanol. Then we mount that into a critical point dryer, which allows us to air dry the sample without causing any air drying artifacts. Air drying artifacts would be shrinkage, cracking, breaking, turning to dust. Then we mount that on a stub, coat it with a heavy metal, gold or gold palladium, that then can go into the high vacuum of the electron microscope and be imaged. The goal here is that we want this picture, when it's in the high vacuum of an electron microscope and being bombarded with an electron beam, to look exactly as it did when it was alive and happy. So what motivated me to produce this image or submit this image was the natural high contrast images of SEM. I just always love the silky and lush overall look of them. They remind me of the paintings of the golden age of the Dutch masters of the 17th century. And when I was playing around with Photoshop, the coloring of this, it just produced an image that to me just looked like something that was painted by ages and ages ago. So Back to showing the image again, and on today, the first day of spring, I think we just say a little nod to our friend the Arabidopsis. It's a humble little plant with a noble contribution to science. Thank you.